Hi, let's continue our discussion on NK models of fitness landscapes. So, we will pick up from where we had left off in the last lecture that an NK fitness landscape can be estimates DNA first of all as a binary string of zeros and ones, and then if this is a DNA of length, let me not call it L but N, then fitness of this DNA sequence can be defined as relative contribution, as the summation of relative contributions from each of the nucleotides associated with this sequence. So, the total fitness is just F1 plus F2 going all the way up to Fn. So, this is pretty straightforward, but what this model allows you to do is that in addition to this, it gives you a tunable parameter which is called K. So, remember these are called NK models. This is where the N comes from, and the tunable parameter is called K. And we will try and understand that what does this represent. And what this represents is something which is experimentally observed very frequently in fitness landscapes, which is called epistasis. Very often when you are experimentally trying to determine DNA or protein fitness landscapes, you will notice that, so NK model will say that fitness for a particular sequence is the sum of Fi, I going from 1 to L, and this will play out to F1 plus F2 going all the way up to F of n. But what you will find is that the contribution that F2 makes will be often dependent on, so let us say this is the second position, this nucleotide makes a contribution of F2, but the contribution that F2 makes will often be dependent on what is present at the ith location. If the ith location or the ith position on DNA in our framework is a 0, F2 might be a certain value, but if the ith position is a 1, F2 will take a different value. So, for instance, let us say the relative contribution of F2 towards the fitness of this DNA string is dependent on what is present on the ith location. If the ith location is a 0, then the contribution of F2, contribution of the second nucleotide here, which let us say is equal to 1, then the contribution of this 1 is alpha. But if the ith location has a 1, then the contribution of that same 1 present on the second position is a beta, and alpha is not equal to beta. This phenomena, that the fitness contribution of a particular nucleotide or loci on the DNA is dependent on what is present elsewhere on the DNA, is called epistasis, and it is a widely observed phenomena when it comes to fitness landscapes. And that leads us to the tunable parameter K, which is associated with NK models. And what K allows us to say, what K represents is, so again for sake of clarity, let us redraw this is length n, and fitness is equal to sigma Fi, I going from 1 to n, this is equal to F1 plus F2 plus Fn. What the tunable parameter K represents is that F1 will obviously, the relative contribution to the total fitness that comes from the first nucleotide is called F1, and F1 will obviously be dependent on the nucleotide I'll call this a nucleotide even though we are dealing with binary sequences 0 or 1. F1 will obviously depend on the nucleotide present on the second position. I'm sorry, F1 is the first position, so on the first position. But in addition to that, what the parameter k represents is that it allows you to incorporate k other positions which determine the contribution k1. Contribution F1. So, what this says is that the contribution that comes from the first nucleotide is not just dependent on what nucleotide is present on the first position, but is also dependent on k other positions elsewhere on this piece of DNA. So, these are k other nucleotides located, these could be located anywhere on the piece of DNA that we are talking about. Together, these k plus the nucleotide whose contribution we are talking about here, all these put together determine what is going to be the quantity F1. So, the relative contribution of the first nucleotide is not just dependent on which nucleotide is present on the first position, but also dependent on these k other nucleotides which are present elsewhere on the chromosome or the string of DNA that you are talking about. That, and you can tune k and incorporate epistasis, because now this is our first step towards understanding how do we incorporate epistasis in our fitness landscapes. And epistasis, as I had mentioned, is a widely observed phenomena in all fitness landscapes that we observe. What this allows us to do is make the fitness contribution from the first nucleotide contingent upon what are the nucleotides present at other locations on the piece of DNA, and hence incorporate epistasis into our fitness landscape models. What happens when I choose k to be equal to 0? Think about this special case for a little bit. We are saying that the fitness contribution of a particular nucleotide is dependent on what nucleotide is present at that location that I am talking about. So, if I am talking about contribution of the first nucleotide, it obviously depends on what is that first nucleotide, is it a 0 or is it a 1, plus it is also dependent on k other nucleotides which could be present elsewhere on the chromosome. Now, what if k is equal to 0? If k is equal to 0, what I am saying is that the fitness contribution of the first nucleotide is dependent on itself and no other positions on the chromosome. So, this is only dependent on itself, no epistasis. So, k equal to 0 is a special case which says that, again that same picture, the contribution of the first nucleotide fitness is equal to sigma Fi, I going from 1 to n equals F1 plus F2 and so on and so forth plus Fl, and this is only dependent on the nucleotide at position 1. Okay. So, that's what's represented by k equal to 0. If that's the case, then how do I determine the fitness of a sequence, of a DNA sequence? What I define is that the fitness contribution of a 0 is, let us say, 1, and fitness contribution of a 1 is, let us say, 2. These are the contributions that I choose for my DNA sequence, and then if I have a DNA sequence which is equal to 1 0 1 1 0. I have this relationship that fitness is equal to contribution of each of these positions. So, fitness of 1, since k is equal to 0, fitness of this 1 in the DNA sequence is only dependent on its own nucleotide and on none of the other nucleotides present on the DNA, because k is equal to 0. Hence, I know that the fitness contribution of a 1 is 2, then I have a 0, and because k is equal to 0, the fitness contribution of 0 depends only on itself, and fitness contribution of a 0 is defined to be 1, and then another 2 and another 2 and another 1. So, fitness of this DNA sequence will be 8 in an NK model with these fitness contributions and with a k equal to 0. So, that is how you would define the fitness for each of the for every possible nucleotide sequence of length 5 here, which will be the number of sequences here will be 2 to the power 5 and not 4 to the power 5, because we are treating DNA as a binary polymer. So, you find the fitness corresponding to each of the, this is 32, each of the 32 sequences, and then connect, represent, do the same exercise that we have been talking about, draw the nodes, draw the edges, connect them if they can be changed into one another via a single mutation, and understand the structure associated with the fitness landscape that you get. So, this k is equal to 0 is a fitness landscape which you get with no epistasis. Okay. What happens if we incorporate epistasis? So, let us again choose k equal to 1. What that implies that the fitness contribution of a nucleotide depends on that nucleotide which is itself, and also one more position, one more nucleotide located elsewhere. So, in this case the fitness is equal to sigma Fi, which is F1 plus F2 and so on and so forth, going up to f of n, if n is the length of the DNA that I am interested in. But now F1 is not just dependent on what is the nucleotide that is present on the first position, on this string, but is also dependent on one more DNA nucleotide elsewhere on the chromosome. So, if I have a DNA string such as this is position number 1. For sake of simplicity, I will define that the F1 contribution is dependent on what is present on position number 1, and that second position elsewhere on the chromosome, which the fitness contribution of this is decided by, is just the neighboring nucleotide. So, F1 is dependent on what is present as nucleotide 1 and what is present as nucleotide 2. So, F1 is a function of nucleotides 1 and 2. Similarly, F2, which is the relative contribution to fitness from the second nucleotide, will be dependent on nucleotides 2 and 3, its neighbors. So, these two go together to determine the fitness contribution from this base, these two go together to determine the fitness contribution from this nucleotide, and so on and so forth. So, we need rules to define this. Since fitness contribution of each is dependent on two nucleotides now, I am going to have to define that the fitness contribution whenever you have a 0 0 is, let us say, 0, whenever you have a 0 1 it is 1, whenever you have a 1 0 it is 1, and whenever you have a 1 1 you have 2. If that is my definition and the sequence that I am interested in is 1 0 1 1 0, then the fitness of this sequence can be defined as, when I look at the fitness contribution of this first nucleotide 1, that is not solely decided by itself because k is equal to 1, that is decided by this 1 and the neighboring nucleotide 1 0, and contribution of 1 0 is equal to 1. When I look at the contribution from the second nucleotide, that is a 0, that is decided by this 0 and the neighboring nucleotide, which is 1, and contribution of a 0 1 is 1. Similarly, contribution for this third nucleotide will be decided by this 1 1, which is 2. For the fourth one will be decided by the sequence 1 0, which is 1, and for the last one, since this is a 0 and no other nucleotide follows this one, we go back to the first one and decide that the contribution of this 0 will be decided by this 0 1 sequence, and that is equal to, according to our definition, equal to 1. This is equal to 6 now. So, in this way you can define k equal to 1 and then come up with nucleotide and therefore define the fitness associated with this particular sequence, having chosen k equal to 1 and incorporated epistasis into your fitness landscape. This again, the length of this sequence that we have been looking at is equal to 5, and hence just as in the previous case, in fact this is the same sequence, just as in the previous case, there are going to be 32 sequences of length L equal to 5, and then what you do here is use this relationship, these assignments of relative contributions of f 0 0, 1 0, 0 1 and 1 1 to find the corresponding fitnesses of all 32 sequences, and then draw the edges and nodes the same way that we have been talking about, and get the corresponding fitness landscape when there is epistasis incorporated into the model. And you can go to higher complexities, you can increase epistasis in the model by choosing higher values of k. So, the maximum possible epistasis that you can incorporate here is when contribution of one nucleotide depends on every other nucleotide on the DNA strand that you are looking at. If that is the case, then every single nucleotide's fitness contribution is dependent on the entire DNA sequence, and that is the maximum amount of epistasis that you can introduce in your model. Okay. We have somewhat randomly chosen two aspects according to the definition that we have talked are chosen somewhat randomly. The first one, if we look here on our slide for k equal to 1, is these fitness definitions. These are somewhat randomly chosen. So, this is aspect 1, and the second one is that when I choose k equal to 1, what I am trying to say is that fitness contribution of the first nucleotide depends on itself and any one of the other n minus 1 nucleotides. Somewhat randomly, I have chosen that one other nucleotide to be the next one for every nucleotide in my system. So this choice of this one other nucleotide which determines the fitness of a particular nucleotide is the second aspect which is somewhat randomly chosen in our definition here. But the point that I want to emphasize here, that these definitions, the NK models, do not claim to define fitness landscape of a particular protein or a DNA sequence in its precise detail. But what they do allow us to do is visualize and analyze the properties of fitness landscape in general by giving us these mathematical structures which allow us to define these fitness landscapes mathematically and do further analysis on. What they also help us with is they give us this tunable parameter k, and for instance, if we know experimentally that one particular system has a large degree of epistasis associated with it, then we can try and capture the properties associated with the fitness landscape of that particular experiment by choosing an NK model with a somewhat higher value of k. On the other hand, if we know experimentally that this system does not seem to have very much epistasis, then we can use the same NK model, but choose a k value to be 0 or 1 and try and capture the properties associated with the fitness landscape of the system that we are trying to study experimentally. And this is important because, as we have seen, trying to determine fitness landscapes experimentally is a very complex task, which is often beyond the realms of experimental constraints. One more thing about these landscapes that we want to discuss, the NK model, is that what sort of fitness landscapes do they give us? So we have, when we have k equal to 0, which represents the case of no epistasis, the fitness landscape that we will get will be a very smooth one, okay. Single peaked and a smooth fitness landscape. So, this is sequence space, this is fitness. As I increase k, as we saw from the slides in the lecture 2 lectures back, we could have smooth fitness landscapes or we could have rugged fitness landscapes. And as we increase k, as we introduce epistasis into our model, we get into these rugged fitness structures where we have multiple peaks, and this will be the fitness landscape associated with increased value of k. Okay. I want to emphasize that fitness landscapes are high dimensional representations, and you can't sort of plot them on a piece of paper. So, these are just cartoon representations of sequence spaces in one dimension. In reality, these are L dimensional objects which are impossible to represent on paper. Okay, so just sort of concluding this idea of fitness landscapes and epistasis, this idea of fitness landscape was first proposed in the 1930s by someone called Sewall Wright. And he had proposed this as a mathematical concept, and it's really in the last few decades that people have, that experimentation has gotten to such a degree of sophistication that allows us to capture the structure of these fitness landscapes experimentally. Let's continue our discussion on these binary sequences, because these help clarify ideas about fitness landscapes and give us some very useful insights into what could happen when populations are moving on these landscapes. And one of the quantities that we are going to make use of is what is called Hamming distance. Again, we are going to be treating DNA as a binary string. So, DNA is just a sequence of zeros and ones, and if we have two sequences both of length L, let us call them sequence I and sequence J. The Hamming distance between these two sequences is represented by a variable called Hij. So, this is Hamming distance between sequence I and J is equal to number of positions where sequence I and J differ from each other. Okay. So, if you are talking of a sequence of length L, all you are interested in when you are computing Hamming distance between two sequences. So, by definition Hamming distance exists between two distinct species, you do not have a Hamming distance for one sequence. Hamming distance, when you are trying to quantify this Hamming distance between these two sequences, they should be of equal length L. Then what you do, all you do is compare base by base every position in these two sequences and count the number of instances where the bases incorporated in these sequences are distinct. So, let us help get clarified with the help of an example. So, let us choose sequence I to be 1 0 1 0 1 0, and let us choose sequence j to be equal to 1 1 0 0 1 0, and the Hamming distance between these two sequences Hij is just equal to the number of positions where the two sequences have different nucleotides incorporated in them. Okay. So, all we do regarding that is first position is the same. So, this does not count, this second position is distinct. So, Hamming distance 1, third position is distinct, Hamming distance 1, fourth, fifth, sixth positions are identical. So, the Hamming distance between these two positions, these two sequences I and j is equal to 2. That is the definition of a Hamming distance. Again, I want to emphasize that Hamming distance is only defined for sequences of equal length, and you need two sequences to be able to compare. Hence, these two subscripts here represent the two distinct sequences of equal length that we are trying to compare and compute the Hamming distance for. Now, when there is replication happening, what is the probability that sequence I was replicating and because there is a finite mutation rate associated with the process of replication, the progeny that was produced was actually sequence J. What is the probability that this happens given that the mutation rate is equal to u, which is probability that a base is copied incorrectly is u, and the length of the sequence is L, and the Hamming distance between I and j, let us say, is equal to alpha. If these things are given to you, that the probability when the sequence is being replicated makes a mistake at a particular base is u, length of the two sequences that we are trying to compare is L, and the Hamming distance between these two sequences I and j is alpha, what is the probability that when I replicates, what you end up as progeny is actually sequence j? I would suggest that you try and work this yourself, and we will pick the next lecture up starting with this calculation itself. Let us stop this lecture for now. Thank you.